Okay, so, hello everyone, my name is Catalina Garcia, and I'm doing my presentation on cell cycle regulation. So let's begin. So the cell cycle, as we know, is an integral part of life. It is both proliferation, which is the rapid division, and then apoptosis. This intricate coupling of these two things is essential for the cell growth and health of the organism. Before we go on and talk about how the cell cycle is regulated, we first are going to talk about what the cell cycle is. Then we're going to go into the regulation, and then we're going to go ahead and see what happens when the regulatory pathways are disrupted. So, okay. This presentation will be broken up into 3 parts to kind of keep our information labeled correctly. The first part will be the cell cycle, and the learning goals for this part will be to be able to describe the cell cycle, and then describe the checkpoints, which we'll talk about, and then the function of the checkpoints. Next will be part 2, which is the regulation. This is what regulation is, how it's carried out by CDKs, which we'll go further into, and then what's being monitored at these regulation checkpoints that we'll see. And then part 3 will be what happens when the regulation doesn't occur, and this can be diseases and cancer, which is what we'll see in a bit. So here we see the cell cycle, and I know it's a lot of text, but really it can all be encompassed, and this is my little laser pointer, so it can all be encompassed by this diagram here. And so first we have the S phase, which we see here, and then the S phase will be the synthesis. This is where DNA is replicated, and then where the DNA is replicated for the daughter cells that will be produced. Then we have the G2 phase, which is the gap between divisions. This is where the new proteins are synthesized, and then also this is where the cell is going to double in size approximately. Then we have the M phase, which is mitosis, as we know, and here it's labeled, and the description here is just kind of a summary of what's happening. So the maternal nuclear envelope is broken, paired chromosomes are pulled to the opposite sides of the cell. Then we have each new set of the daughter chromatids are then going to be surrounded by new nuclear envelopes as they're getting ready to divide. And then here we see in cytokinesis is where cell division does occur as the cell is pinched in half and then 2 new daughter cells are produced. So then we kind of go into this, and then we do go into the G1 phase. This is after mitosis. So at this point, the cell can decide to either continue dividing again one more time, or it can go into a G0 phase, which is where it's not going to continue on. And so as we see here, it states that rapidly proliferating tissue or embryonic tissue will continue to divide. It will split again after this G1 waiting phase, but if it does cease, it enters, as I said, the G0 quiescent phase. This phase may last hours because in this phase, the cell can continue to divide if it does decide. So this phase can last a couple hours, or the cell can decide to not divide any further, and then this G0 phase will be for the rest of the cell's life, basically. And so we see here just a visual of that represented. And this slide is what we're going to see is kind of the timestamp, which I thought was very interesting on how each of these sections, how long it's going to take. So we see in the S phase 6 to 8 hours, and then in the G2 phase 3 to 4, and the M phase, the G1 phase, and then the G1 phase being 6 to 12 hours. And then that G0 phase, as we see here, it could be for the rest of the cell life. So in this little section, it says terminally differentiated cells withdraw from cell cycle indefinitely. So the cell's deciding not to divide any further. But again, as I stated earlier, if the cell does decide to continue dividing, it will be re-entering at the G1 phase. And so you see here, re-entry point. Okay. So continuing on, we're still in part 1, the cell cycle. I just included this slide because I want to talk more about those gap phases, because they're not just phases in which the cell is at a standstill. The cell is active. And so in these phases, we see that they're meant to separate the S phase from the M phase. So the S phase, as we know, is where DNA is replicated, and the M phase, as we know, is where the chromosomes are segregated. And then, so that's what's important about the gap phases, they're separating those two phases. And then, as I stated, these aren't just inactive cycles, but this is where the cells can gain, integrate growth signals, mass, and then organize the replicated genome and prepare for chromosome splitting. So this is just to state that those two gap phases are important and integral to the cell. So continuing on. What are checkpoints? Checkpoints are very vital in the cell cycle and especially in the health of the cell. So checkpoints are essential in the forward process of the cell cycle. And what this means, as we can see by this little picture here, is that throughout the, here, let me go back. We'll see another visual earlier, but throughout this phase, there are checkpoints that if the cell doesn't meet these checkpoints, then the cell cycle will be halted. And so what this little picture here is showing is that, see, all violators must fix their mistakes or face apoptosis. So when those checkpoints are not cleared, the cell can either fix whatever is happening, or it can decide like, hey, this might be too much to fix. Let's go ahead and do the programmed cell death. And so that's why these little guys are kind of nervous, because they're either going to fix their mistakes or they're going to go through apoptosis. So what are checkpoints? What are checkpoints surveilling? They're surveillance mechanisms. They're monitoring the order, the integrity, the fidelity of the major events in the cell cycle, which may include growth to the appropriate cell size. So this is kind of what they're checking in the cell. That the replication and integrity of the chromosomes are well, and that the segregation at mitosis is accurate and all going well. So again, just to state that the checkpoints, if they're not passed, then the cell cycle will be halted. The cell will deal with whatever violation is occurring, or it's going to go into apoptosis because, well, we'll go on to that actually, why apoptosis occurs. So we'll leave that there for now. So that was the end of part 1. Now we're going to part 2, which is the regulation of the cell cycle, and then here we'll see by CDKs and cyclins. So via Nelson and Cox 2017, we have this information. The timing of the cell cycle, as we stated, is regulated by a family of protein kinases. So the activities of these kinases are changed in response to the cellular signals throughout the cell cycle. They are heterodimers, and they consist of a cyclin, which is a regulatory subunit. This is the activator, and then a cyclin-dependent protein kinase, which is the CDK. So this is the catalytic subunit, which we'll be breaking down. Catalytic. So without the cyclin, the CDK is inactive, so it needs to be bound for the CDK's activity to rise. As we see here, it says when the two do bind, the activity increases, and I did not state here, but it increases by 1,000-fold because it was inactive, and then it goes into it. So we have a quote here. Vanden Heuvel, 2005, gives us, the balance between CDK activation and inactivation determines whether cells proceed through G1 into S phase and from G2 to M through regulatory mechanisms that are conserved in more complex eukaryotes. So this is kind of just restating what I had stated earlier, just a better interpretation of it, that these checkpoints, the CDK activation, will make sure that the cell cycle isn't just dividing and going with any violations or with any mutations. So it states here, it will decide whether the cells proceed into the different phases of the cell cycle. So in this next picture, we're going to see, okay, so we have a more abstract visual of the cell cycle and the cyclin and CDK pairings that will encompass these sections. And here we can see that they're different ones. So cyclin D, CDK 4 and 6, cyclin E, CDK 2, and so on and so on. Just showing that there are other checkpoints. It's not just one checkpoint. There's a variety of things going on in the cell cycle to make sure that it's regulated correctly. And so these are just, the CDK and cyclin couplings are a big aspect of that regulation. And so right now we're going to see apoptosis, which is the other very important aspect in cell cycle regulation. So we see here why kill a cell. Vermeulen et al. 2003 states that apoptosis and proliferation are intimately coupled, which I stated earlier. So here we can see how they're coupled. The linkage of cell division and programmed cell death can be recognized by, so they're linked by these following genes: CDK, cyclin, CKI, as well as PRB, P53, and others. The fate of the cell is likely due to the interplay of these mentioned genes. So it's not just one factor into apoptosis, but it's a lot of interplay between these guys here as to whether apoptosis will occur. So continuing on with that, here we see kind of the conditions in which apoptosis is even presented as an option. This would be adverse growth conditions. Complex signal transaction networks are initiated when those growth conditions are found. And then the information is processed and sent to subcellular organelles. So it's not just kind of like a one and done kind of, okay, apoptosis now, this is wrong. The signal transaction network is initiated and the information is processed to other portions of the cell. So further, we have a nice quote here by Vermeulen, which states, "The specific pathway chosen depends upon a variety of damage. The presence of functional P21 and its crosstalk with PRB and the genetic background of the cell." So it's not just one aspect like we stated earlier. The decision of apoptosis is a variety of signals and this is all to ensure that the cell is making the right decision to halt this cell's life. And again, we have our little picture showing that either the cell fixes what's wrong or apoptosis will occur. So yes, let's keep going. So that was part 2, that was regulation. We had the CDK and cyclins, and then the apoptosis when things go wrong, and the CDKs, find something that's beyond repair, we'll say. So what does happen when these regulatory pathways are disturbed? Here we have oncogenes. Oncogenes are mutated versions of the genes encoding signaling proteins. So this is a big problem in the cell because of how they're involved in cell cycle regulation. One of the main aspects of this is that the mutations will continually give the cell okay to divide, which can cause tumors. So as we saw earlier, the cell is going through all these checkpoints and it's making sure everything is neat and tidy and everything that's supposed to happen is happening. But when these oncogenes are just telling the cell to divide, these checkpoints are not being regulated. They're not being involved in the decision to replicate. So remember, as we stated earlier, when a problem does occur, the cell cycle is halted. And so the cell cycle stops, and then the cell, by all these signal transactions, decides, okay, wait, can we fix this? If not, apoptosis, because as you saw, the process is a long time. And we can thank our body that we're not just kind of going through with these mutations. So they can lead to tumors. These oncogenes can affect a lot of areas of the cell, so encode defective receptors, G proteins, protein kinases, which we saw earlier are the family of CDKs, which if that were to be, as we see, if that's affected, like that's a huge effect on the cell. And then you can see that oncogenes can also encode defective growth factors. So there's a lot of ways that the regulatory pathways can be disturbed. Okay, continuing on. So we have the oncogenes, and then we have tumors. And so this happens when tumor suppressor genes encode the regulatory proteins that normally inhibit the cell division. And then these mutations do result in tumor formation. So cancer is generally caused from an accumulation. Remember, it's not just one thing. So it's the accumulation of the mutations in the tumor suppressor genes as well as the oncogenes, which we saw oncogenes can be a variety of things. Okay, so cell cycle deregulation is also a common feature of human cancer. And we say human cancers because we'll see later on that the cell cycle differentiates. Cancer cells often proliferate unscheduled, which we saw earlier, that they just have that rapid proliferation, that rapid dividing without accounting for the checkpoints. So cancer cells often proliferate unscheduled, which displays frequent genomic instability and chromosomal instability. So here we see what is genomic instability. Genomic instability is the increased DNA mutation as well as chromosomal aberrations, which is a huge part of the cell cycle. And then the chromosomal instability is changes in the chromosome number. And we see that when the change in the chromosome numbers happen, we get diseases such as Down syndrome. We get a lot of those embedded mutations, which affect the whole genome. Okay, continuing on. Okay, cool. So here, okay, I can zoom in. Okay. Here we have a comparison of the yeast, mammal, and cancer cycle. And I find this very interesting because you get to kind of see exactly what's different here. So first we have the yeast, which is it looks, very similar, pretty much the same to mammals, which we would be in this category here. We have G1, we have S, we have G2, we have mitosis, and then, so that's yeast. And then here we have a classical model of the mammals. Again, we have S phase, G2, M phase, and G1. This is interphase specialization. So then here also we see the cyclins. So we see cyclin E, we see cyclin A, we see cyclin B, cyclin D, and these are coupling with the CDKs. So we have the CDKs here. You see 4 being in use here. Here at the evolutionary conservation, this is the essential cell cycle. We also have that S phase, G2, M phase, although they're not broken up as you see here that they're broken up. M phase and G1 phase. Again, we see that the cyclins are in use. Remember those regulation. These are the activators for the CDK. And so here we can note that there's only one CDK in use via this picture. And then here we have the specialized cell cycles. So we have S phase, G2, M phase, G1. We have cyclin, and then we have some more CDKs that are paired with it. Okay, so these are more or less similar, different things happening based on, the specialization that we can see here. We would be in these categories. And then here we have the cancer one, which is very interesting because, it's the very same foundation as we know. So S phase, G2, M phase, G1. And then we have CDK1, where here we don't see any cyclins being used. So this is aberrant mitogenic signaling. This is when, as we saw earlier in the previous slide, that the unscheduled proliferation, so the cell is just dividing, it's ignoring those checkpoints, or those checkpoints are mutated, as we saw, because cancer is that combination of the mutated oncogenes and the tumor suppressor, what was that again? Let me see. Tumor suppressor genes. And so see here it states that cancer is the accumulation of these mutations and the tumor suppressor genes and the oncogenes. So back to this, we see unscheduled proliferation, as we stated, and so active mitogenic sensors, defective mitogenic breaks, and then overcome oncogenic stress. Then we have genomic instability, which we stated earlier. This was a huge, one of the main factors. So we have defective DNA repair and DNA damage checkpoints. So remember, those checkpoints are what's really important in the regulation of the cell cycle, because in those checkpoints, you guys probably already know, that's when the cell is halted because something's abnormal. And then the cell decides whether to continue on, fix the problem, continue on, or go through with apoptosis. So next we have chromosomal instability, which we did state earlier as well. And this is the defects in mitotic checkpoints and chromosome segregation. So when those chromosomes aren't segregated, then you have two daughter cells that are not going to have parallel parallel chromosomes, the chromatids aren't going to be equal. And so again, we see that the mitotic checkpoints aren't being regulated. So you guys probably remember, I can go back to it. Let me see. So I'm going back to this picture here. So these checkpoints, what I was just saying is that these checkpoints aren't being regulated either, which is a big deal because we know our cell cycle is essential in, dividing. So continuing on here. Okay. So yes, we have our cancer cell. We see what's kind of going on. We see what's going wrong. We see how the foundation is very similar to our essential cell cycle, but we do see that these are mutations and things just that aren't right. And because of this unscheduled proliferation, we're getting a lot of division in cells that are just replicating very fast. Okay. Okay, so just a review of what we went over. The cell cycle, very important in the maturity and growth of organisms. Like, this is the growth of organism. It is the proliferation of these cells. And these cells have a very detailed and intricate phases that each have distinct functions. So we have our S phase, we have our G1, we have our M phase, we have our G2, and then we have our G1. So each of these steps are essential and they all do important steps. So as we stated earlier, those gap phases between S phase and M phase, they're not just phases of inactivity, but during this time, the cell is going through a lot of growth, a lot of organization, it's getting ready to split. And so these intricate steps need to have checkpoints, because so much is going on that, our bodies are amazing because so much is going on, yet we have these regulatory functions to make sure that each step is, that each step is being monitored and everything that's going on is appropriate and not abnormal. So we have those checkpoints. As we see, those checkpoints are performed by a family of protein kinases. This is those CDK and cyclin couplings that we saw in the cell cycle earlier in the picture right before this. This regulation is necessary for healthy cell proliferation. And again, I just want to state that cell regulation is the cell cycle checkpoints and proliferation, but also the apoptosis. So continuing on, so just stating again that these regulatory factors, apoptosis, the CDKs and cyclin couplings, is to ensure that abnormal progression of the cell doesn't happen, which we see in cancer when the cell's just replicating so much, and other threatening diseases such as leukemia and other such occurrences can happen if regulation of the cell cycle is not being listened to, which happens in cancer. So that's the cell cycle, that's the regulation. Yes, okay, so I think we'll end on this great picture of, the regulatory factors as well as what happens when those regulations are not listened. Thank you.